Each of our cells contains our entire genetic heritage. However, while all of our cells contain the same information, they obviously don't use this information in the same way. A skin cell is nothing like a neuron, and a liver cell doesn't have the same functions as a heart cell. What's more, twins who share the same genome are never perfectly identical, like a Regina pizza from one restaurant to another. In these examples, the explanation for this particularity is called epigenetics. As we saw earlier, and under certain conditions, some recipes are not used because they are hidden. In a restaurant, the menu can change according to the weather. For example, hot, hearty soups are out of fashion in summer. The study of the epigenome corresponds to the study of changes in access to the different recipes. Even though they do not involve variations in the DNA sequence, these changes can be transmitted when the cell divides. As opposed to variations affecting the DNA sequence, the very text of the recipes in the book, epigenetic modifications can be reversible. The epigenome thus consists of marks on the genome, like pieces of repositionable adhesive tape that will cover or uncover recipes to indicate when and where the recipe will be accessible or not. Epigenetics is focused on an additional layer of information that defines how these genes will be used or not by a cell. A gene is then said to be active, switched on, or inactive, switched off. If these epigenetic modifications persist, even when the signal that induced them disappears, this can lead to a genetic disease. The study of the epigenome, in other words, the analysis of the distribution of these marks throughout the genome, can reveal genetic diseases for which there is no DNA modification, but rather there are epigenetic modifications. The epigenome can also reveal genetic diseases that involve DNA modification, which results in a characteristic epigenetic profile. But it's not only genetic factors that cause epigenetic modifications. The environment, such as diet, smoking or stress, can also lead to epigenetic modifications, that is, the switching off of genes. In the near future, other examinations are likely to contribute to our understanding of rare diseases that have yet to be elucidated, the ones for which we do not yet know the recipe, the gene that is lacking. These examinations include proteome and metabolome analyses.